Hello, my name is Alina Goyle, and I'll be presenting my research on the effect of Filaggrin mutations on the development of atopic diseases. To start, the term atopy refers to the genetic inclination to produce a hypersensitive immune reaction in response to common allergens. The prevalence of atopic diseases has been increasing in recent decades, and currently around 20% of the world's population has at least one atopic disease. The four predominant atopic diseases are eczema, asthma, allergic rhinitis, and food allergy, and all are characterized by an inflammatory response affecting different parts of the body. Eczema affects the skin, asthma the lower respiratory tract, allergic rhinitis the nasal mucosa, and symptoms of food allergy can manifest in different locations depending on the person. The atopic march refers to the progression of atopic diseases, typically starting with eczema in infancy and progressing to food allergy, asthma, and allergic rhinitis later in childhood and during adulthood, depicted in this diagram here. Though the mechanisms causing the atopic march are not completely understood, theories suggest that the damage to the epidermal skin barrier as a result of eczema may cause the allergic sensitization that leads to the development of the subsequent atopic diseases. Filaggrin is a gene that plays a key role in skin barrier function, encoding proteins that prevent transepidermal water loss and the entry of allergens. Around 10% of the European population has a filaggrin mutation, making it pretty prevalent. Null or loss-of-function mutations in filaggrin are one of the strongest risk factors for eczema, and two of the most common filaggrin loss-of-function mutations in European populations, 22A2del4 and R501X, have both been shown to be strong predisposing factors for eczema, along with two other less prevalent mutations, S3247X and R2447X. However, the effect of filaggrin mutations on other atopic diseases has not been widely studied. Therefore, my study aimed to investigate the effect of filaggrin loss-of-function mutations on the development of eczema, as well as allergic rhinitis, asthma, and food allergy. In addition, the sub-goal of the study was to determine if filaggrin mutations affect the age at which an atopic disease develops. The study population consisted of the fair birth cohort, which contained 969 children born in the Isle of Wight between 2001 and 2002. Children were assessed at ages 1, 2, 3, and 10, and phenotypic, environmental, genetic, and epigenetic data was collected. Of the 969 children, only 295 had genotype information available, leading to a total of 35 children with any of the four filaggrin mutations. The specific breakdown of the individual mutations is shown here. In terms of methodology, we define the outcome of disease as having that disease over at least two consecutive data points to prevent potential one-time misdiagnosis from affecting the results. Atopy was defined as having an outcome of two or more atopic diseases. We ultimately did not analyze the individual effects of S3247X and R2447X mutations due to their low prevalence, being present in only 3 individuals each. All analysis was performed with Microsoft Excel and Jupyter Notebook, and a p-value of less than 0.05 was considered significant. We hypothesized that the duration of breastfeeding, gender, mode of delivery, so vaginal or cesarean, and only child status may have confounding effects based on results from previous studies, and therefore we analyzed their effect. We used a logistic regression model to determine the association between duration of breastfeeding and the development of each atopic disease. This test was chosen because the dependent variable, outcome of disease, was binary, while the independent variable, duration of breastfeeding, was continuous. We found that duration of breastfeeding did not have a significant association with the development of any of the atopic diseases at any age point, shown in this table. Correlations between the independent variables, gender, type of delivery, and only child status, with the dependent variable, outcome of disease, were analyzed with 2x2 contingency tables and Fisher's exact test, as both variables being tested were binary and the sample size was under 1,000. Type of delivery and gender were not significantly associated with any atopic disease. However, only child status was significantly associated with eczema at age 1. The associations between filaggrin mutations and disease were stratified into 4 analyses, presented here in the flowchart. We examined the overall effect of filaggrin mutations in general on the outcome of diseases, as well as at each specific age point. In addition, we tested the effect of the 2 most prevalent filaggrin mutations individually on the outcome of diseases and the diseases at each age point. Once again, using 2x2 contingency tables and Fisher's exact test, it was determined that filaggrin mutations do not impact the outcome of atopy, nor the outcomes of each atopic disease. The p-values, odds ratios, and 95% confidence intervals are shown here. Next, we analyze the effect of filaggrin mutations on the development of each atopic disease at each age point, so ages 1, 2, 3, and 10. Significantly, filaggrin mutations were associated with eczema at ages 1 and 2, and allergic rhinitis at age 3, and food allergy at age 10. Also of note is that filaggrin mutations had no significant effect on the development of asthma. In the third analysis, neither the 22A2del4 nor the R501X mutations had a significant association with the outcome of atopy or any of the atopic diseases, p-value shown in the chart. Finally, looking at the effect of the individual filaggrin mutations on atopic diseases at each age point, we found that the 22A2del4 mutation was not associated with eczema, asthma, or food allergy at any age. However, there was a significant association between that mutation and allergic rhinitis at age 2, but not at any of the other ages. Similarly, no association was found between the presence of the R501X null mutation and eczema, allergic rhinitis, or asthma at any age. The mutation was strongly associated with food allergy only at age 10. Interestingly, the variables considered confounders in previous studies did not have a significant effect on the outcome of atopic disease in this study. Conversely, the strong effects of filaggrin mutations on eczema at ages 1 and 2 supported results of prior studies that found significant correlations between eczema and filaggrin mutations. Our study found inconclusive and variable results about the effect of filaggrin mutations on allergic rhinitis and food allergy. However, the absence of any association between filaggrin mutations and asthma in any of our analyses is also of note because it validates the results of previous studies that state that filaggrin mutations do not affect the development of asthma. To conclude, our lack of strong correlations between filaggrin mutations and the outcomes of eczema, rhinitis, and food allergy suggests that our definition of outcome may have been too restrictive, as it only included participants with at least 2 consecutive instances of disease. In addition, the inconclusive effects of allergic rhinitis and food allergy may stem from the low number of participants with a filaggrin mutation, as well as with each individual mutation studied. Future research would test our findings in a larger scale study to ascertain the effects of filaggrin mutations more accurately and to concentrate on the S3247X and R2447X mutations that were not individually analyzed here. I would like to thank my mentor, my science research teachers, and my classmates for their aid in my project. And here are the sources I referred to.